Okay, we're going to start lecture 13. We are still in module number 2, talking about cell biology and histology. Our learning objectives for this lesson is nucleus and protein synthesis and characteristics of cell biology. All right, so protein synthesis, this occurs in 2 steps. Transcription is when the DNA information coded into a messenger RNA, and translation is when messenger RNA is decoded to assemble polypeptides. So let's put that in a picture here. We have the information flow during protein synthesis. First, we have DNA that is going to go down through RNA processing, and it's going to turn it into pre-mRNA or messenger RNA. It's going to process again, and it's going to turn it into messenger RNA. That process is called transcription. It's going to leave the cell and go out into the cytoplasm, and it's going to undergo translation. Okay, that's where it's going to become a polypeptide. It's going to extract all that out and become polypeptides. Genetic code. So each 3-base sequence on DNA, which is the triplet code, is represented by a complementary 3-base sequence on RNA called a codon. And then the genes, these are segments of DNA that carries instructions for creating a protein chain. All right, the central dogma theory. So this is a dogma of molecular biology that states that DNA contains instructions for making a protein, which are copied by RNA. RNA then uses the instructions to make a protein. So in short, we have DNA transcribes to go to RNA. So we have DNA goes through transcription to make RNA, which goes through translation to make a protein. Or DNA to RNA to protein. That's the central dogma theory. Okay, here's a little bit more detailed version. Protein synthesis steps. Step 1, the DNA unzips and opens up the nucleotide base letters, the base code. The nucleotide base code is read by the RNA and converted to messenger RNA by transcription, which creates a new copy. That messenger RNA leaves the nucleus, goes to the ribosomes. The messenger RNA nucleotide code is translated by transfer RNA. Matching up the nucleotides are brought in, 3 linked together is going to be an amino acid. The amino acids linked together is a protein. On the slideshow, you can watch this video. All right, DNA nucleotides, when they're going to pair up, A is going to go with T, which is adenine and thymine. Cytosine is going to go with guanine. So anytime you see those base pairs, the A is going to bond with the T, the C is going to bond with the G. It's always going to be that way. Where RNA nucleotides, the A, adenine, goes with uracil instead of thymine, because in RNA there is no thymine. It's subbed out with uracil. And then cytosine is always going to go with guanine as well. All right, the roles of RNA in protein synthesis. So RNA is pretty much the go-between molecule or the communication molecule that links DNA to proteins. RNA is going to copy the DNA code in the nucleus, then it's going to carry it into the cytoplasm to the ribosomes. All RNA is formed in the nucleus, and RNA differs from DNA. Like we said before, the uracil is substituted for the thymine, and RNA has ribose instead of deoxyribose. So that's where the R for the RNA comes from, ribose nucleic acid, and DNA is deoxyribose nucleic acid. All right, the roles of the messenger, ribosomal, and transfer RNA. So there's three types of RNA. Messenger, single-stranded, code from DNA template strand is copied with complementary base pairs, resulting in a strand of messenger RNA. This is called transcription. Ribosomal RNA is a structural component of ribosomes, the organelle where protein synthesis occurs. Along with tRNA, this helps to translate the message from messenger RNA into a peptide. And then transfer RNA, or tRNA. This carries the amino acid, has special areas that contain a special triplet code or anticodon that allows each transfer RNA to carry only a specific amino acid. The anticodon of tRNA will complementary base pair with codon messenger RNA at the ribosome, adding its specific amino acid to growing peptide chain. This process is referred to as translation. All right, and last but not least for this section is autophagy and apoptosis. So autophagy is cells that have become obsolete or damaged need to be taken out of the system. These are going to be more like organelles that get taken out. Apoptosis, or also known as programmed cell death, this causes certain cells like cancer cells, infected cells, old cells to neatly self-destruct. It's like they kind of just eat themselves, self-eating. And that's going to be it for this slideshow. If you have any questions, please let me know.